Hello students, welcome to this video where we are going to do this topic of gluconeogenesis. Very important video, must do video. So let's start. First understand the name gluconeogenesis. Genesis means synthesis. Gluco stands for glucose. Neo stands for new glucose. So it is synthesis of new glucose. Why we are referring to new glucose? Because it is not the glucose coming in diet. It is new glucose getting synthesized in body from non-carbohydrate sources. Right? Obviously, this pathway will occur in fasting or starvation when body is not eating food, right? So glucose not coming from outside, and glucose is a very important fuel for most of the cells. So that's why it is important to synthesize it, right? Now there is a rule of 2 for gluconeogenesis. Rule of 2, easy to learn. It occurs in two organs, liver and kidney, right? Then it occurs in two compartments of the cell. Glucose synthesis uses two compartments in the cell. One is mitochondria. It starts from mitochondria. Then it occurs in cytoplasm. So both mitochondria and cytoplasm are used. Then it occurs in two situations. One situation is not eating food. Means not in fed state, means the body is either in fasting or starvation. So when we are not eating food, either in fasting or starvation, then gluconeogenesis will occur. Then second is diabetes. Diabetic situation, I told you, is same like fasting situation. So in diabetes also gluconeogenesis occur, because although there is too much of glucose in diabetes, there is hyperglycemia. Too much of glucose is there in blood, but the peripheral cells of the body, most of the cells of the body, they are not receiving glucose. So body thinks I am in fasting. That's why in diabetes gluconeogenesis will occur. Very important point, right? Then there is a concept that you have to see what happens to link reaction in fed and fasting state, and then you will understand something for gluconeogenesis. So let me take that. Okay. So in fed state, there is hormone insulin. Insulin activates link reaction. So what will happen to link reaction? That this link reaction, pyruvate to acetyl-CoA, will occur. The presence of enzyme PDH, pyruvate dehydrogenase, right? But in fasting state, what will happen to link reaction? Let's see on next slide. In fasting or starvation, there occurs beta oxidation of fatty acid. There is breakage of fats from adipose tissue. Fatty acids are released via blood, they reach liver. In liver, there is beta oxidation of fatty acid. The end product of this beta oxidation is acetyl-CoA. And in fasting, there is excess of this acetyl-CoA present. Okay. So what will happen to link reaction? Link reaction is pyruvate to acetyl-CoA, and the product of link reaction is already present in excess. So this excess acetyl-CoA product will itself inhibit its enzyme and link reaction will stop occurring. So we already know that link reaction is kind of anabolic reaction. It occurs in fed state and not in fasting state. So link reaction will not occur, and the substrate of the link reaction, pyruvate, will start accumulating. So this pyruvate will be diverted for the formation of oxaloacetate. Okay. See, pyruvate is a 3-carbon compound. Oxaloacetate is a 4-carbon compound. So we need to add carbon dioxide here. So name of enzyme will be pyruvate carboxylase. pyruvate carboxylase, right? Now, we already know that this acetyl-CoA activates pyruvate carboxylase, which is the first step of gluconeogenesis, right? And be careful here that acetyl-CoA is never glucogenic. It cannot make glucose. It is not the substrate for glucose, right? But acetyl-CoA activates the very first step of gluconeogenesis, that is pyruvate carboxylase, conversion of pyruvate to acetyl, conversion of pyruvate to oxaloacetate, right? Now, what is glycolysis? Glycolysis is glucose to pyruvate. Glucose to 2 molecules of pyruvate, okay, because glucose is 6 carbon, pyruvate is 3 carbon, right? And what is gluconeogenesis? Gluconeogenesis is pyruvate, the main substrate is pyruvate, we can say, pyruvate to glucose, obviously 2 pyruvate will be taken to make 1 glucose, right? So can you see it is reversal, it is glucose to pyruvate, this is pyruvate to glucose. So now if I ask you a question that is gluconeogenesis just a reversal of glycolysis? Is gluconeogenesis just a reversal? Then your answer is no. No, it is not just the reversal because 3 enzymes are different. See, glycolysis pathway has irreversible enzymes. There are 3 irreversible enzymes. So, instead of these irreversible enzymes, we have to use different enzymes in gluconeogenesis. So, 3 are different, but other 7 will be same. Okay, there are total 10 in glycolysis. So, 3 are different, another 10, so 3 are different, another 7 will be same, right? So, let's see on next slide which are these 3 enzymes. So, in glycolysis, the irreversible enzymes are pyruvate kinase, PFK1 and hexokinase, right? Now, which enzyme instead of these will be used in gluconeogenesis? Okay, so instead of pyruvate kinase, there is use of 2 different enzymes in gluconeogenesis. The first one is pyruvate carboxylase, next is PEPCK, phosphoenol pyruvate carboxykinase, right? Then instead of PFK1, there is fructose 1,6 bisphosphatase, okay, right? Then instead of hexokinase, in gluconeogenesis, we use glucose 6 phosphatase, right? So these are the 4 special enzymes of gluconeogenesis. Very, very important to learn these, right? As you do this pathway, they will be automatically getting learned, right? Just go conceptual and you will understand. So now let's see the reactions in cytoplasm and mitochondria, the reactions of gluconeogenesis. Okay, I'm going to make this diagram. So first I will be showing cytoplasm and mitochondria here. Okay, so here in cytoplasm, glucose gets converted to pyruvate, right? There is one step of pyruvate kinase from phosphoenol pyruvate. Okay, so glucose gets converted to phosphoenol pyruvate in glycolysis. This is glycolysis occurring in cytoplasm, right? There is this enzyme pyruvate kinase, the last enzyme of glycolysis, which forms pyruvate in cytoplasm. Okay, now let's see how gluconeogenesis will occur, right? In gluconeogenesis, this pyruvate will enter mitochondria, right? You know that this is pyruvate proton symport with the help of which pyruvate enters mitochondria, right? Now, you already know that the first step of gluconeogenesis is formation of oxaloacetate from pyruvate. Enzyme is pyruvate carboxylase. Any carboxylase uses ATP, right? ATP is used here, carbon dioxide is added here, right? Now only this step occurs in mitochondria. Now next steps will occur in cytoplasm. Okay, so this oxaloacetate has to come out of cytoplasm, has to come out of mitochondria. So I'm writing short form of oxaloacetate as OAA, right? So oxaloacetate is coming outside. Now oxaloacetate has to come outside because other reactions will occur in cytoplasm, right? So, but oxaloacetate cannot cross inner mitochondrial membrane. So how this will happen? First, oxaloacetate will be converted to malate. Malate can cross inner mitochondrial membrane and reaches cytoplasm. In cytoplasm, malate can be again converted to oxaloacetate, right? And here NADH is used from oxaloacetate to malate, and from malate to oxaloacetate NADH is produced. So tell me, is it malate shuttle? We have done shuttles in the one of the previous video. So tell me, is it malate shuttle? No, this is not malate shuttle, right? Here NADH is getting used, it is getting used in mitochondria, and we are getting NADH in cytoplasm, right? But the purpose of shuttle is different. Shuttles take NADH from cytoplasm to mitochondria so that it can take part in ETC, right? So here what is happening is that we want to take oxaloacetate outside, right? We are not taking care of what is happening to NADH here, right? Just the purpose is to bring oxaloacetate outside from mitochondria to cytoplasm, right? So this was one of the special enzyme pyruvate carboxylase. Now next let's see what happens. Oxaloacetate will be converted to phosphoenol pyruvate. Okay, so here oxaloacetate is 4-carbon compound. Phosphoenol pyruvate, it's a 3-carbon but with a phosphate. Okay, so from 4 to 3 carbon there is removal of carbon dioxide, right? And for addition of phosphate here, GTP is used in this step. GTP is used, right? This is very, very important step. Name of enzyme is PEPCK. Full form is phosphoenol pyruvate carboxykinase. Okay, the name of this enzyme is very, very different. Okay, that see actually decarboxylation is occurring, but they are writing the name as carboxy, right? So this is the second important enzyme of gluconeogenesis, right? And now can you note here that because pyruvate kinase is irreversible, so from pyruvate to phosphoenol pyruvate we cannot simply go like this. Okay, we cannot just reverse this step. So what we have done, pyruvate going into mitochondria, making oxaloacetate, then oxaloacetate getting converted to phosphoenol pyruvate. Okay, so we have followed this route instead of this route, right? So instead of pyruvate kinase, these two enzymes, pyruvate carboxylase and PEPCK, these two are involved, right? Now next we have to reverse this from phosphoenol pyruvate to glucose. So let's have a look on next slide. So let's see this detail on the next slide. So now have a look at this slide. Here I am showing glycolysis pathway only, right? So we have reached till this step in gluconeogenesis, we have reached till this step of phosphoenol pyruvate, right? Now we have to go in this opposite direction till glucose. Okay, so let's see. The enzymes which are reversible enzymes of glycolysis, they are same in gluconeogenesis also. So enolase, phosphoglyceromutase, phosphoglycerokinase, glyceraldehyde 3-phosphate dehydrogenase, this isomerase, aldolase A, they all are same in gluconeogenesis also. Okay, so this is very important. They ask you that which enzyme is common to both glycolysis and gluconeogenesis. So be careful about this, right? So these are enzymes which are common. Okay, then next enzyme is irreversible enzyme of glycolysis, that is PFK1. So in opposite direction we have a different enzyme here. What is that enzyme? See, what is happening is that now substrate is fructose 1,6 bisphosphate. and the product is fructose 6-phosphate. So what is happening here? What is happening? There is removal of phosphate from position number 1. Okay. So the enzyme is phosphatase for the removal of phosphate. So to tell the complete name of enzyme, take the name of substrate. Now substrate is fructose 1,6-bisphosphate. So enzyme is fructose 1,6-bisphosphatase. Okay, that is the enzyme of gluconeogenesis. Then next is also reversible step. So it is same in gluconeogenesis, right? The next step is hexokinase step, which is irreversible. Now in opposite direction, we have to convert glucose 6-phosphate to glucose. Again, this is phosphatase. It is glucose 6-phosphatase. Okay, right? So now let's revise the 4 enzymes of gluconeogenesis. The first one is pyruvate carboxylase, which is present in mitochondria. Second one is PepCK, which is present in cytoplasm. Third is fructose 1,6-bisphosphatase, which is present in cytoplasm. Fourth is glucose 6-phosphatase. This is not present in cytoplasm, because if you keep this enzyme in cytoplasm, then in cytoplasm glycolysis has to occur, and glycolysis wants to convert glucose to glucose 6-phosphate. Only then this pathway can occur, right? So this enzyme will reverse this reaction. Okay, glucose 6-phosphatase will reverse this reaction. So that's why this enzyme is not kept in cytoplasm. This enzyme is present in endoplasmic reticulum, in the membrane of endoplasmic reticulum. So let's have a look on next slide. So let's say this is cytoplasm, right? And this is endoplasmic reticulum. This is membrane of endoplasmic reticulum. Here we have, in the membrane, we have this enzyme glucose 6-phosphatase, right? So what happens that in cytoplasm our gluconeogenesis is occurring, most of the steps, and in the end we get glucose 6-phosphate. Okay, this glucose 6-phosphate will not be broken down in cytoplasm. It will go into endoplasmic reticulum. This glucose 6-phosphate, with the help of a transporter, it will go into the endoplasmic reticulum lumen. So this is lumen of endoplasmic reticulum, right? Here this reaction will occur with the help of this enzyme. So this enzyme will break it down into glucose plus inorganic phosphate, right? Then this glucose can come out with the transporter help. Inorganic phosphate also can come out. Okay, so this way it happens. So there are 3 transporters here in the membrane of endoplasmic reticulum. This one is T1 transporter, which is for the entry of glucose 6-phosphate, right? This is T2 transporter, which is for the exit of glucose. This is T3 transporter, which is for the exit of inorganic phosphate, right? All these transporters obviously are active during fasting or starvation, when this pathway of gluconeogenesis is occurring, right? Then next let's do substrates of gluconeogenesis. Very, very important topic, frequently asked. Okay, so first is pyruvate. We just did the pathway, it started from pyruvate only, right? And note one point that mostly 3 carbon compounds are good substrates for gluconeogenesis, right? So this pyruvate, it is also 3 carbon compound. Then second is lactate. Lactate is also a 3 carbon compound. Lactate can be converted to pyruvate, and then pyruvate can form glucose, right? Then third is glycerol. Glycerol is also a 3 carbon alcohol, right? Then fourth is propionic acid, which is derived from odd chain fatty acids. This is also 3 carbon, right? Then fifth is amino acids. Amino acids. Which amino acids? Glucogenic amino acids and both category amino acid, which are both glucogenic and ketogenic. Okay. There are 18 such amino acids which can make glucose. Out of these, most glucogenic is alanine. Most glucogenic is alanine. Alanine is a 3 carbon amino acid, right? So you have seen mostly 3 carbon compounds are good substrates of gluconeogenesis, right? Then sixth is any TCA intermediate. Any TCA intermediate. Okay. I told you acetyl-CoA is not the intermediate of TCA, but all others like citrate, alpha-ketoglutarate, succinyl-CoA, fumarate, they all are substrates. They all are intermediates of TCA cycle, because any intermediate of TCA cycle can make oxaloacetate, right? It makes oxaloacetate only, right, in the end. And we have seen in the pathway of gluconeogenesis that oxaloacetate is one of the intermediate here and it can be converted to glucose. So any TCA intermediate is glucogenic. Then now, next let's learn that which amino acid is glucogenic, which is ketogenic and which is in both category. Okay, I will help you learn this thing very easily. So amino acids, any amino acid which in the end, in the catabolism of that amino acid, in the end, if we get acetyl-CoA or ketone body, if this is formed in the end of catabolism of amino acid, then that amino acid is considered ketogenic, right? We have only 2 ketogenic amino acids, very easy to learn because both starts from L, right? One is leucine, other is lysine. Okay. Then any amino acid which forms pyruvate in the end, or some TCA intermediate in the end, then that amino acid is considered glucogenic. Okay. And we also have some amino acids which are both glucogenic and ketogenic. Okay. So how to learn these? See, in ketogenic both starts from L. In both category, take a mnemonic TIP. Okay. From T, write down all the amino acids starting from T. That is threonine, tyrosine, tryptophan. Okay. Then I stands for isoleucine. Okay. P stands for phenylalanine. Okay. So take this mnemonic tip, TIP tip. From T, write down all the amino acids starting from T. I is isoleucine, P is phenylalanine. So these 5 amino acids are in both category. We have 2 amino acids in ketogenic category. So how many are left out of 20? 13 are left. 13 amino acids are left, right? So 13 are purely glucogenic, right? And I already told you in this most glucogenic is alanine. So glucogenic are 13, purely glucogenic are 13, purely ketogenic are 2, and in both category we have 5 amino acids. Okay. So tell me in total how many are ketogenic? In total how many ketogenic? 2 plus 5 is 7. 7 amino acids are ketogenic, right? And in total how many will be glucogenic? How many is glucogenic? 13 plus 5. 13 plus 5 is 18. So in total 18 amino acids can make glucose, right? Only 2 amino acids cannot make glucose, that is leucine and lysine. Then these 2 amino acids, leucine and lysine, I told you they are ketogenic. But this lysine is in controversy. Okay, just consider it in controversy. Some books say different, some other books say different thing. So be very sure about leucine. If question is asked and both are given, mark leucine as ketogenic, right? Because there is a confusion for lysine, right? Whenever there is a controversy, just note down it is a controversy, and depending upon the question, you will be easily able to solve if you know that this is a controversy, right? Then now in the end, let's do energetics of gluconeogenesis, right? So which enzymes are using glucose? So which enzymes are using ATP or GTP here? First enzyme, pyruvate carboxylase, that uses ATP, right? Then PepCK, next enzyme PepCK, I told you it uses GTP to add the phosphate, right? Carboxylase uses ATP because all carboxylases uses ATP, right? Then there is one enzyme PG kinase which is involved in glycolysis also. It is involved in gluconeogenesis also, right? It is the common enzyme. This enzyme uses ATP because in glycolysis this enzyme produces ATP, okay, by substrate level phosphorylation, okay. In gluconeogenesis opposite will happen, ATP will be used. So these all are used here, be careful. Usually in pathways we write that this much ATP is produced, okay, but gluconeogenesis is energy consuming pathway. It uses energy to make glucose, right? It is high energy consuming pathway, okay. So for calculating energetics we will be using these 3 points only, right? Let me be very clear here that if NADH is produced in some reaction or in some pathway, then we will count it as 2.5 ATP produced, right? But if in some reaction or pathway NADH is used, then do not consider this thing for any energetics, okay? Because NADH is present free in the environment, it can be used. So do not count this for any energetics whenever NADH is used. Do not count. Whenever NADH is produced, then though we count only, right? So if one pyruvate is used, then these 3, 2 ATP and 1 GTP is used, okay? But you know that for making one glucose, 2 pyruvates are used, right? So it will be 4 ATP and 2 GTP used to make one glucose, or you can say 6 high energy bonds are used to make one glucose. So can you see, when we do aerobic pathway of glycolysis, we get 7 ATPs, right? And if we are making glucose via gluconeogenesis, then 6 high energy bonds are used, okay? Then pyruvate to glucose, 6 high energy bonds are used, okay? Then lactate and alanine are also very common substrates for gluconeogenesis. Again, 2 lactate and 2 alanine will be used. Lactate can be converted to pyruvate, alanine can be converted to pyruvate. There is no counting of energetics here from lactate to pyruvate or from alanine to pyruvate, okay. So if they ask you lactate to glucose, how many ATPs or high energy bonds are used? Here also answer will be 6, right? From alanine to glucose, how many are used? Again 6 high energy bonds, or to be very precise, 4 ATPs and 2 GTPs will be used, right? And this is it with this video. Thanks for watching and best wishes to you.